by this, I invite Dr. Stefano Panzeri to the stage.
Thank you for your introduction, and thank you also for your patience. We had some technical problems here because of the setup of the intranet here, so I apologize for this. It took some time to understand the problem, but also I wanted to understand, also given the time change and so on, how much time do I have for the talk? Just want to make sure I don't go over time. So could you tell me how much time do I have for the talk?
We have 45 minutes for your talk.
Okay, so this should be comfortable. So hopefully I make it 35 minutes, so we recuperate a bit the delay. And thank you for the invitation. Thanks all to the Sharif people. It's a great pleasure and honor for me to take part in this symposium. And I would like to tell you a bit more about my research here that we do here in Hamburg in collaboration with various labs about the possible causal and functional role of the correlation between activity of different neurons in shaping some important brain functions, such as the encoding and the readout of sensory information. And before I go on, I would like to thank my collaborators, especially the students, the postdoc from my lab who did all the computational work, that are Monica Moroni, Giulio Bondanelli, Eugenio Pasini, Jacopo Bonato, and Human Safai, that I think studied the Sharif years ago, and he worked with me for a number of years and did some important contribution to this. And I would like also to talk the collaborating laboratories, especially that of Chris Harvey, where most of the data that I present here were collected, and also those of Dima Rimberg at New York University. And our contribution to the work I'm going to present is the computational one, so develop methods and ideas and interpret the results, but the empirical data were collected by my close collaborators here. And so the problem we're going to study, as I told in the title, is the contributions of neuron-to-neuron interaction to functions such as sensation, perception, and decision-making. And so we study what sometimes, especially in magnetic functional resonance imaging, fMRI, is called functional connectivity, or also sometimes noise correlations and so on. But essentially what it is the correlation between the activity of different neurons, so their activity in terms of action potential to spikes. And here, unlike, for example, studies with fMRI, the study at the coarser scale, we really focus at the level of population of neurons and neuron-to-neuron interactions. And why this level is important, I think it's obvious, because of course there are many important level organizations in the brain, but definitely for most function, what happens is that groups of neurons essentially encode that information and then transmit to other neurons for other computation. So this is definitely a level which is crucial to understand the brain computations and brain functions. And the question we try to address is first about the role of this interaction between neurons to the encoding of sensory information. And also the second one is how the functional interactions contribute in a causal way, so in a direct way, to creating not only information encoded inside the brain, but also creating perception, so what we can perceive, or also informing behavioral choices that we have to make, in response to the sensory evidence that we have. And the functional correlations, I guess some of you also come from physics, and the reason why those pairwise functional correlations are important to study understanding their role is because those are essentially the key ingredient, the first step toward understanding the emergent properties of a population of neurons. By emergent properties, I mean the properties that emerge from the system as a whole, so from the interaction between the elements, in this case the neurons, and this is some property that you cannot understand by looking at one element at a time or by neglecting the interaction between the elements. So that's why I think it's important, and it has fascinated people for many years. And the kind of task that we use to address the role of population of neurons in encoding sensory informations and then use this encoded information to generate a perceptor or to take a decision is what is called a perceptual decision-making task. Essentially, what a perceptual decision-making task is you present some visual or whatever, auditory and so on, some sensory stimulus. So you give inputs to the sensory parts of the brain. This translates into neural activity into some region of the brain. And then this neural activity encodes for the information from the senses. So all what your brain knows about is in this activity of these neurons. You don't have access directly to the senses. You have access to your own neurons. And then based on the information and the neurons encoding their activity, you have to generate, so if you have a sensation, you essentially, you perceive, so you have a perception. And one way to know whether or not you perceive something, you perceive it correctly, is to ask the subject to generate a behavioral report of their percept. For example, if I see a piece of cheese, I eat, or if I, for example, as in the case of the task that we consider, if I hear a sound from the left, I turn toward the direction of the sound, and if I hear a sound from the right, I turn toward the other direction, so always toward the direction of the sound. That's a way to essentially evaluate whether or not your percept is forming correctly, and also how you use those sensory signals to take appropriate decisions. And the decisions here are appropriate because turning left when the sound from the left gives the animal a reward, so they get their water or whatever it is. And when they don't go into the right direction, so their percept is incorrect, they don't get a reward. So that's why it's useful to understand how activity in the brain generates appropriate decisions and perceptions. And in this study that we studied, my colleagues at Harvard, this Harvey Lab, they train mice to run through a linear maze in virtual reality while being on a styrofoam ball. And while rotating the styrofoam ball while running, they were essentially running through this virtual environment, was a virtual linear maze. And while they were running, they heard the sound from the left and the right, and they turned at the end of the maze toward the direction of sound to collect the reward. And while mice were playing this video game, essentially, we were recording the activity of their neurons in the superficial layers of posterior parietal cortex using two photon microscopy. So two photon microscopy uses fluorescent images of neurons. This fluorescent indicator captures changes in calcium concentration. Those are related to spiking activities. From those you can essentially reconstruct the spiking activity of the neuron with reasonable precision. This is called calcium imaging, and the reason why this was done in posterior parietal cortex is that it's a so-called association area, so an area which essentially is thought by various evidence to transform sensory information into behavioral choices. So it's a very good question, a very good brain area to address the question that we are posing. And we recorded about 50 to 100 neurons simultaneously in each session. And since those neurons were recorded simultaneously, we can compute the correlation between their activity. So correlation between neural activity is simple function that you know very well, and then quantifies how the activity of neuron 1 in the trial depends on the activity of neuron 2 in the same trial. And this is really a within trial correlation, and the most known measure is the one that we discussed today, is so-called noise correlation. So the correlation computed in trial with the same sensory stimulus, and that's done to discount the covariation due to same tuning or different tuning to different stimuli. So those are correlation, are called noise because they are correlation of activity that, being at fixed stimulus, do not reflect tuning to the stimuli. They just reflect essentially processes that are not related to the stimulus. And one cartoon one way people like conceptualize in neuroscience those plots is to plot scatter plots of the activity of the neurons in each trial. So in this cartoon, for example, you would have two different stimuli, for example, sound left and sound right, colored by different colors. And each dot is the joint activity in the two-dimensional space, so the joint activity of a neuron in a given trial, and each ellipse is the stimulus-specific distribution. So distribution to stimulus 1 with pink, and distribution to stimulus 2 with blue. And if they are correlated, you have an elongated shape. Elongated shape, it means that I can predict the activity of neuron 1 by the activity of neuron 2, because when neuron 1 is particularly active in response to the stimulus, neuron 2 will also be particularly active, and also when neuron 1 is particularly not active, so silent, neuron 2 will also be predictably particularly silent. And if you have correlations like this, then you have an elongated cigar, a banana, and an ellipse. If they are not correlated, so if at fixed stimulus I cannot predict one neuron to the other, then it means that they are uncorrelated. You get in this pictorial representation round distributions. And in the brain, every time you put an electrode or a microscope and you record neurons, you get a situation like this. So neurons talk to each other, they are correlated. So definitely there are emergent properties, and not consider one neuron in isolation from the rest. And what we're going to do is to study essentially the function of this interaction between those neurons. And the first function we consider is the one that is traditionally considered in my field of neural population coding, and it is the one about encoding information. So essentially, it's the relationship between the stimuli that have out there, in our case, the sound from the left or from the right, and the patterns of neural activity. This is called sensory encoding, and it's an important function because it's an important step in order then to use this information to take decisions and to generate perception. If you don't have that, then you know nothing about the external environment. And so it's very important to understand this. And but I will argue in the talk that although it is very important, it is not enough to study this, and I will try to explain why. But also I wanted to say something, that if you at any time wish to interrupt me because you have questions and something is not clear, please do. I'm very happy to take questions anytime. And also I'm aware that when there is an interdisciplinary audience, it's very difficult to all give all the relevant information. So if something is unclear or if you have a comment, please feel free to interrupt anytime. So first, I will present very shortly result that we have about the encoding of information. So how the correlation, the functional interaction between those different neurons shape the encoding of sensory information. So how much information I have about what happens in the external worlds in the pattern of neural activity. And the analytical approaches that we and others over the years developed to study this, can be also very complicated if you want, but conceptually are very simple. And here I will present them in the most simple conceptual way. And essentially, the idea is to first build decoders of neural population activity that we use to reconstruct the stimulus based on the pattern of neural population activity in each trial. And here I did a cartoon from a previous paper of us. So this is again this space of multidimensional neural activity where each dimension is the firing of each neuron. Here I plot only 2 neurons, but you can imagine this, of course, a multidimensional space. If you have 50 neurons, there will be a 50-dimensional space and so on. What you do, you draw essentially a boundary. For example, this is a linear decoder that best separates the stimulus-specific distributions when you train your algorithm. And then if the data fall in the right part of the boundary, then you decode the stimulus correctly, and so you can reconstruct the stimulus is correct. The decoding will not always be correct from a small population because neural activity is noisy, but in this way you can quantify in a simple way the information that the neuron population encodes about the stimulus. And then if you want to know what's the role of functional correlations, you can easily destroy those correlations, for example, by trial shuffling, when you create pseudo-population neural response vector where you associate responses to the same stimulus for each neuron but coming from different trials. Then you build surrogate data that have exactly the same single neuron properties, but where there are no more those correlations. So by comparing information in both cases, sorry for the problem with the slide, by comparing information in both cases, then you can understand if correlations have some information that you cannot access from the original data, if the correlations are not good for information encoding. And we have studied this, also many people studied this for many years. And to cut a very long story short, sometimes you have that information is enhanced by the presence of this correlation because they vary with the stimuli. But in the vast majority of studies that I've seen, and also in the study that I present today, this doesn't happen. What happens is exactly the contrary. So what happens is that correlations decrease the amount of information that you have in neural activity. You would have more information if you didn't have correlation, but the same thing on neuron properties. And the cartoon is something like this, because this is the case without correlations. And what you see here is the overlap between those two distributions of neural population activity to different stimuli. When they are separated, it means a point like this, I can decode it. If I see activity like this, I decode it correctly as belonging to the blue stimulus. If I see activity here, I can decode correctly because it belongs to the orange stimulus. But here in this overlap, I cannot decode correctly. So the bigger the overlap, the less information I have. And if you look at this cartoon, when you have correlation, so when you elongate those axes, what you have is that you increase the overlap between those specific distributions. So more often than not, what you have is that when you shuffle the data, so when you destroy correlations, you have less information encoded. You have more information encoded in neural activity than in the original data, where you have the same single cell properties, but no correlations. So what happens in most cases, and also what happens in this data, is that correlations decrease the amount of information in neural activity, and so it's a bad thing. It's not a good thing. Ideally, you would have to have as much information as possible. And because of this negative result, many people over the year, there were many influential papers, for example, from Bill Newsom, from Alex Pouget, many nice papers about this. Because of this result, that I said, we found it in our data set, but it's found many times in the literature. But because of this, people led to the conclusion that correlations are bad for your perception, because they decrease the information, so they limit performance in forming accurate perception discrimination, and accurate behavioral choice and behavioral responses. And that's, I think, it's the conclusion that many studies have reached. But so my point of view, I think, is, if this is really bad for you, why do you have them? Why every time you put your electrodes or microscope to look at brain activity, you find correlation between the neurons? If they're bad for you. Evolution put pressure, so your function should be highly optimized in order to survive. And so we thought that maybe something is missing from this picture. And another evidence that something is missing from this picture came from doing a very simple empirical reasoning. And the empirical reasoning is as follows. So if correlation, so as I said, in this task, we made deliberately the task difficult, so that sometimes the animal, most of the time, like 70-80% of the time, the animal does it correctly, but sometimes the animal does it incorrectly. So the animal is unable to use this information in neural activity about the sensory world to perceive correctly and then to take the appropriate decision given the perception. And so we can look at correlations between neurons, between correct trials where the animal decided correctly and error trials where the mouse decided incorrectly. And if correlations are really bad for you, then you would expect higher values of correlations in error trials and lower values in correct trials. I think it's apparent why. But if we look at the real data, with various measures here, pairwise and population-wise and so on, we found exactly the opposite. When the behavior is correct, you have stronger correlations than when behavior is incorrect. So it's the other way around. You have less information in neural activity because of stronger correlation, but somehow those seems to be beneficial for behavior because they are stronger in correct than the error trial. So when we found this result, our first impression was, this is paradoxical, because this is exactly the opposite you would expect from those established theories. And so, we thought about it, and we thought that maybe the reason why they are good for behavior, even if they encode sensory information, is that they help the downstream propagation of sensor information, and they help to better inform behavior. So maybe you have less information, but it is most or better used because of those correlations. And so to kind of investigate this, we develop a measure that we call readout, which is the mathematical relationship between sensor information expressed in neural activity in each trial and the behavioral report that the animal makes in each trial. And why I feel that this is a measure of the causal impact of the information neural activity on downstream areas and behavior is that okay, we don't record all the intermediate steps between this pattern of activity and behavior, but we record what is essentially ultimately the final output of all this post-processing on neural activity, and the final output is the choice. So essentially this measure is useful because it recapitulates the overall downstream causal effects of feature of neural activity on behavior. So readout for us is a map between the sensor information expressed by neural activity in each trial that we know to be decreased by correlation and the behavioral choice. And given that we had better behavioral performance with stronger correlation, we thought that essentially this map, this readout of information from patterns of neural activity would somehow work better when information is more correlated or more consistent across neurons. And to do that, as I said, we developed this map, this mathematical function that I will explain in the next slide, which is essentially a map between sensor information, neural activity, and behavioral choice. And we first consider the readout that is implicitly assumed or sometimes even explicitly in all these previous studies of neural code. So the previous studies of neural code looked only at the information encoded in neural activity, and that would explain your perception, your decision, under the assumption that you somehow are able to follow the optimal computation. In that case, the more information you have with an optimal computation, like a Bayesian computation and so on, so the more information you have available, if you're optimal, the better you can do it. And so first we examine what would happen with this readout. And here we make a cartoon where, again, we have this correlated activity, which is like the real data, and then this is the correlated activity when you remove correlation because you shuffle the trial. And as I said, if you have something like this, having correlations reduces the information. So correlation are bad for the information encoded. And then if you assume that you have an optimal computation, then the probability that we plot here with this color map that the choice follows the neural activity depends only on the stimulus which is decoded. So from population activity, so the result of the Bayesian output. So if activity is here, it will be with some probability reflected in stimulus blue. If activity falls here, will be stimulus yellow, and so on and so on. So if we have a map like this, a readout that only reflects the output of the optical computation, then if you simulate choices like this, you would have that behavioral performance will be proportional to the encoded information. So lower behavioral performance with correlations when you shuffle away the correlations. But also this would predict something that goes directly against our data, and that is that you would have, as I explained, because correlations are bad, higher correlation strength between the neurons in error trials than in correct trials, which is again exactly the opposite of our data. Then to find something that will be more consistent with our data, we made the assumption that the readout utilizes the redundancy. So you can extract more information if the population that you have, in this case, in this cartoon too, but you will have more in the data, provide consistent stimulus information. So if both neurons, both population essentially report, decode the same stimulus. So they both decode stimulus 1 or they both decode stimulus 2. And mathematically, the way to represent this map, so this probability that the choice follows the indications of the stimulus encoded neural activity, is that this is higher when the two neurons decode the same stimulus. Like this, for example, here, both neurons low firing rate, so correlated. And so both indicate stimulus 1, then when they indicate stimulus 2. So essentially our hypothesis that when you are in these regions, consistent information, you have a higher conversion of your bits encoded in the brain, so the stimulus encoded in your activity, into behavioral choice. But when those two neurons give inconsistent information, then it's harder for the downstream circuits to make sense of the evidence and produce a behavioral choice that corresponds to the indication of neural activity. And this is, of course, a suboptimal computation, because the optimal computation is always to follow the Bayesian decoding of the stimulus, so this S hat, so the Bayesian decoder. And but maybe this is not so possible for the downstream system. And one simple example I make about redundancy is, for example, someone having to gain information about physics, hearing an answer from Einstein and another answer from me. And if you are an optimal decoder, you would learn or you would know that you should pay attention to Einstein and maybe not pay attention to me. But if you are a suboptimal decoder, you know nothing about physics, then if by chance I say something that goes against what Einstein said, you will be confused, because you have two contradicting hypotheses, and you will be less able to trust the information, to use the information that you receive. And so this kind of decoding that we propose to explain the data is exactly like this. So it works better, the readout, when it receives consistent information from the neurons, and it works as well when it doesn't get. And if you have something like this, then you would predict that first you will have, because now correlations, so correlations, what they do, they increment the number of decoding errors, but also they increment the proportion of trials that can be read out or utilized correctly, which is this in the white region. So because you utilize better the trials, then you have higher behavioral performance with correlated activity than with uncorrelated activity. And also, like in our data, you have stronger correlations in during correct choices, during correct behavior, than during incorrect behavior. So this suggests that this maybe it's a good idea. And this, until now, is just a sketch, it's just a cartoon, but we try to take this seriously and apply this reasoning to the data. So what we have in the data is that we have in each trial, we have the stimulus that we present that we know, we have the behavioral report of the animal that might or might not match the stimulus that was presented. And also we have the stimulus decoded from neural activity. This is the stimulus decoded from the entire population activity. But also we have the stimulus decoded from each neuron, which can tell us whether the neurons act in a correlated, in a consistent way or not. And so what we do, we produce these results in each trial where we decode optimize the stimulus for neural activity. And then we make a parametric model of choice in every single trial, which is exactly like the one I put in a cartoon. So apart from some terms that are not important empirically, but they are not important now, we have a term which is the one of the Bayesian decoder. So the choice follows the stimulus encoded in population activity, and this would be the only term you have if you were optimal. And then you have a term, essentially an interaction term between the information encoded in neural activity and the consistency of information across neurons. This term would amplify the readout when information is inconsistent, and would instead decrease the strength of the readout of information when the stimulus is inconsistent across neurons. And to cut a long story short, we found that when we wanted to explain the data, so the single trial choices of all trials or mice and so on, you really had to have those terms here to explain the choices well. So these interaction terms existed, and they were exactly like in our cartoon, so that they amplified information, the readout of information, when information was consistent across neurons, with respect to the case when it was not consistent across neurons. And so essentially, we have evidence from the data that the readout that we propose in this cartoon is the one that explains the data. And if you have a good model of sensory encoding, which we have because we can compute it from the data, and you have a good model of information readout, which we have also because We also computed it from the data. Then what you can do is compute the behavioral performance that those neurons would contribute to the amount of discrimination from behavioral performance, given the information encoded in neural activity and given how it is read out. And you could do this either giving the real correlated data or giving the shuffled neural activity, and then see what happens. And if we do this game, we would have the prediction that because of the increasing consistency in neural activity due to correlation, which facilitates the readout, we would expect that those neurons provide higher information for this perceptual discrimination when you have correlation, as in the real data, than when you don't have them. So this would suggest that, as indicated by the fact that correlations are stronger during correct trials, they have a measurable positive impact on behavioral discrimination performances, because maybe they don't help, they damage information encoding, but they make this information more affordable downstream. And also a side product of this is that I think we were able to demonstrate and measure the actual contribution of emergent properties to actual macroscopic functions, so in the spirit of thermodynamics. And here we see really a macroscopic effect. So I don't know, a population of 50 neurons because of this correlation contribute, I don't know, about 0.05% more behavioral discrimination. So of course, it will build up for larger population, but it's a measurable macroscopic effect on behavior of microscopic interaction between the neurons. And of course, there are also caveats with this approach because the fact that we related it to behavior, I think, is suggestive of a causal downstream effect, and that's the way we interpret it. But of course, it's based on observational data. And when you base all your inferences, all your observations, it's all a little bit dangerous because, for example, it could be that those neurons are not those that cause perception behavior, but they are receiving, for example, a copy from the activity of the real neural population that generates behavior. And one way to do this, to essentially make sure that this is not the case, would be to perturb the activity of the neuron during perception. For example, with two-photon optogenetic, where you can shine light on neurons and activate them at will, or push them toward being more active, while we image the activity. And in that case, we would predict that if we get a pattern of stimulation of the brain that, for example, activates all neurons with the same choice preference, left preference, which is this pool of neurons, consistently, then you would have a bigger push of behavior toward left than if you injected the same amount of information, but inconsistently, like in this upper row. So this is low consistency, same amount of information, like a neuron with big information, and you stimulate a neuron with big information, but that prefers left, and you stimulate a neuron with lower information that prefer right. You get intermediate amount of left information. You can get the same amount of information injected by perturbation toward left, stimulating to intermediate neurons. And the prediction is that you have higher behavior readout of this simulation than of this simulation. And we were unable to do this experiment yet because the effects of behavior of stimulation of a few neurons during visual stimulation is quite difficult to achieve. But I think we were able to do some experiment conceptually similar, creating a virtual perception. So we didn't consider the case of altering perception of an existing stimulus, but altering perception of a non-existing stimuli. So my colleagues here at New York University, Di Marimber and colleagues, did an experiment where they trained mice to recognize and get a reward when reporting the presence of a target spatiotemporal stimulation of the olfactory bulb by selecting different parts in the olfactory bulb and activating one at a time. And then the mouse has to detect the presence of optogenetic pattern stimulation in the brain without any other stimulus, either target or non-target, with the same number of items is stimulated at random. And you also have perturbation trial where you just perturb a little bit dose, and by measuring the reports, modeling the reports to the stimulation, the perturbation trials, you kind of can understand the boundaries of perceptions of the stimulus in the spatial neural population activity. So you can compute distances, perceptual distances in the spatial neural population activity. And by doing that, you get a good idea of how different patterns are read out. And what you can do, you can apply exactly the same regression model with the interaction between the activation of different neurons at different times. And if you do that, you find that the model that best explains the relationship between the perceptual reports of the animal and the causally evoked neural activity is very similar to the one that I told you before, with interaction terms between the activation of different populations that have exactly the same structure as the one that we derived observationally from our work. So it's a result which is causally consistent with interaction being key to brain information processing. And I think now I'm getting close to the time limit. I think I have another 2 or 3 minutes. And so I conclude with work that Human Safaya, my postdoc that graduated from university in Tehran and now is completing his doctoral training at Harvard Medical School, but worked with me for a number of years. And in our collaboration, we try to look with further evidence of these correlations at the population level for information transmission in the brain. And one way, I think one nice way to do it is to change a little bit the concept of population code with respect to what always proposed in the literature. And what is proposed in the literature is a concept based on recording neurons that are nearby. So you put a bunch of electrodes or you image the neurons, but you don't know where the neuron projects to. You just look at nearby neurons in a given area. But you can argue that the only information that is transmitted in the brain is from groups of neurons that project to the same anatomical target. For example, here some neurons might project to this area, some other neurons to this area, and so on and so on. And one way to now try to address this with modern techniques is to put a retrograde tracer here. You inject in the anatomical target retrograde tracers. For example, you can inject some blue label in some virus here that travel back the axon and then goes to label those neurons here that you are imaging. So if you do that, you know among, say, the 100 neurons here, which ones project to form a group that project to the same anatomical target. So we can define population codes that project this information to the same anatomical target. And if we do that, we found that within a field of view, so within a spatial region, you have much stronger correlations between neurons that are in the same neighborhood that project to the same anatomical target with respect to randomly selected neurons that were selected in previous studies. So you have a stronger correlation and a stronger decay of correlation between correct and error trial when you consider neurons that project to the same target, which suggests that correlations are really useful for transmitting information. And also another thing that we noted is that not only you have stronger correlations, but you have a non-random network structure only when you consider neurons in the neighborhood that project to the same target. And one way to quantify structure is to quantify triplets, for example, neurons that positive or negative information interaction, so correlation that increase or decrease information. And what we found, to cut a long story short, that in neurons that are close to each other but don't project to the same target, you get a random network, so a randomly organized network. But with neurons that project to the same target, you get a network like this, a network with an excess of those kind of triplets of neurons that essentially are all positive, so same sign, and also a decrease of number of triplets that instead have opposite behavior, like, for example, the product negative sign. So you have an excess of triplets like this that have a plus, so plus, the product of their sign, or also they have plus because of the configuration, so which is minus time plus. So essentially you have a pool of neurons organized in synergistic groups that are connected redundantly. And this structure, you see it only in neurons that project to the same target. And to try to understand what's the function of this structure, we did some expansion of the total information that those population of neurons would carry. And to cut a long story short, we were able to quantify the contribution not only of single neurons and pairwise information and so on, but also about how they are structured in triplets above and beyond the random structure, the random arrangement that may have. And what we get is that by this expansion, the motifs that we found to be in excess with respect to the random network in the data, that are the neuron with positive products of interaction between them, they contribute positively to the information, whereas neurons that have a negative sign of the product of interaction contribute negatively. So now the fact that you had an excess in the information, in the population of triplets, so structure of triplets organized in a way that increases information, it means that this structure that we see only in neurons that protect the same target increases the information that is available to the neuron to form choices. And I think this is conceptually important because it shows that there is a microscope. So the fact that there is a microscopic interactive structure only neurons that boosts this information, only neurons that protect the same target, it means that the kind of constraint or design of the neural code is not a design to encode information locally, but to make it available to other targets to further computation, which is compatible with the other results that we have. And I think now also, since because I started late, I think I'll cut the last part of the talk and go to the conclusions. And the conclusions for me is that our work from the idea that the role of the functional interactions of the emergent properties of neurons in a population can only be reached considering their effect on downstream computations to inform behavior, not only to understand local computations. And so it's not enough to consider local encoding of information, neglecting its downstream consequences. And when you try to instead consider those both issues, our conclusion is that the design of population codes is not to be based only on optimization of local information representations, but on the trade-off between the richness of this local information encoding and its safe transmission to the behavioral output. And also, I think I gave some example toward the end that this conclusion, of course, remain developed reading out, so recording your activity and correlating it in a sophisticated way to behavior, but they can also be tested causally by using intervention to write information in the brain and so to provide causal interpretations to the data. And I think with this, I conclude my talk, and I thank you for your patience and your attention during the talk. Any question you have, we'll be very pleased to address it. Thank you very much. I hope the connection did not.
Thank you very much for the interesting talk. If there is any question, please ask it. So there's a question. Can you hear the voice of the questioner? Please ask it.
I cannot hear any voice. I hear you talking, but I don't hear the question. Maybe would it be possible to repeat it near the microphone? Okay. I don't know if someone could repeat the question, then I'll try to reply. Thank you.
Can you hear me?
Yes.
Can you hear us?
Yes.
Stefano, can you hear us?
I can hear you, but the other voice was distant.
So the question was why? I mean, apparently you were recording from layers 1 to 3. The question was there a specific reason to record from these?
Yes, the reason was that with two photon microscopy, it's easier to record from superficial layers because otherwise, if you go to deeper layers, you have a higher scattering of light. So the reason was technical because it's just easier to record with two photon microscopy. And the reason why it's useful to record with two photon microscopy is that first, it's easier to get larger number of cells, which was critical for our work. And second, then at some point, it's easier to be able to perturb them with optogenetics because you can see the cells, so you can direct the light to them. But okay. Yes, yes. I mean, first, I would say that it's a very good idea because neuroscience and computational neuroscience is a hot topic throughout the world, and it's important for basic research, but also for applications because many of the ideas of computational neuroscience have trickled to artificial intelligence. So it's a very marketable and very useful job. And also, from my point of view, it's very fun and very satisfactory. If you come from physics, for example, I can tell because I think there is a very, I don't know, a very real chance that you can make an impact with your skills, with your thoughts, because the field is new, it's emerging, and it's very important. And also, there is a high chance that you have fun because you can apply, the clever concept that you had the graduate school in a new way and really enjoy the challenge intellectually. So my advice is, try to work hard on it if you like and have fun. And if you have fun and do really good research, you have your reward. And the second that I think it's important for a young person that comes with the numerate background, not only to apply algorithm or models blindly, but try to get a really good understanding of the data, their interpretation, their limitations, and to make sure that you understand well what are the new questions, the most important questions that have not been addressed yet, and also to make sure that your I don't know, your analysis, your interpretation, the knowledge that you get from the data is correct, and you understand them. So always, I would say, look at the data, try to understand them deeply, try to understand deeply the questions of the experimental people, and then from there try to understand what is, um, what can you add? What new computation, new modeling, new algorithm can you add, and new knowledge. So that would be my advice. Sometimes clever computational people start from the algorithm and then try to apply them. And I think it's nice to have your background, your ideas from your numerate science, but I think it's also important to try to understand what the biologists want to know that is not known. And then I think it's easier to make a progress and make an impact. Thank you. Thanks to all for your time. Thank you. Okay.
Hear
And I
us?
see the virtual. Okay.
Can you hear us?
Yes.
Okay. Thank you. So there are a few more questions in the room. My colleague, Dr. Rashid Shamali, and I also have a question. So
Good.
maybe I do my question first and then you do it. So in your talk, you showed that there is a reduction of correlated activity in error trials, which
Yes.
was unexpected. It was very interesting.
Yes.
But what you didn't show was that most times when people do decoding of error
Yeah.
trials, there's also a reduction of the decoding capacity when you do SVM or LDA or any type of
Yes.
classifier. Usually there is a
Yes.
reduction of decoding from the neural population at the same
Yes.
time. So just the fact
Yes.
that the noise correlation has gone down doesn't tell us anything about whether the means of those two conditions might have been closer to each other in error trials. So in that case, then having less noise correlation would be a secondary point. Basically, what I'm trying to ask is that you must have looked at the error trials as well. So in the error trials, do we lose capacity to decode the two conditions? And if the answer is yes, then why do you think the lack of noise correlation is an important thing in that case?
Okay, I think yes, we looked at it, and as you say, I think as you correctly guessed, and is reported in many studies, also the firing rates, so the one-point functions of the neurons also decrease in quality. And so one component of the error trials is that individual neurons are also more noisy. But at the same time, it is true that noise correlations are stronger in correct trials than in error trials. And that, so my understanding, my take of this is that both factors, single neuron properties and correlations, contribute to behavior. And the reason, I think, you might think that it's a secondary contribution, but I would like to argue that it's not. I mean, first, if it was a secondary contribution, it would be important anyway. And second, also, those things scale up differently in terms of population size. So all the theoretical models and also empirical data when are reliable for large population show that, I mean, essentially the contribution of information to information scale linearly or quadratically, it is signal to noise ratio, but it is information linearly with the number of neurons, whereas the contribution of correlation scales quadratically with the number of neurons. So if you consider two neurons, the addition that you have from the interaction between them is, I don't know, 5% or something like this. But if you consider a population of neurons, the limitation that you get because of noise correlations are like 5 times larger than those of individual neurons. There was a paper by Mark Schnitzer in Nature. I think the first author was Rumiantsev a couple of years ago, where they had recording of that size, and it was exactly their conclusion. So that's why I think it's important to understand if there is a contribution also from noise correlation, because those may be small for small population, but it scales up big time. But the second thing is also, I don't think there is a single coding mechanism. I think both like single neuron properties and emergent properties contribute, and I think we have just to document both and try to understand them and make sense of it. So I don't know if that answers your question.
I think that partially answers it, but basically, the reason I'm also asking this is because you suggested that we don't. So, one, we have two frameworks. The normal framework people think about is the ideal observer framework, which works
Yes.
with finding the best border between the two conditions that can
Yes.
separate the two conditions.
Yeah.
But you were kind of proposing an alternative framework of redundant kind of a,
Yes.
you basically showed 4 quadrants of information and stuff like that, which is
Yeah.
kind of new and maybe a bit unconventional. So what I was trying to say is that maybe it's still the system that works is an ideal observer. And of course, it works better in correct trials because the means of the two distributions are further away from each other, even though the two neurons are
I mean, yes.
correlated, but still the classifier can classify them correctly. In the error trials, there's less noise correlation, but there's also the means of the two distributions are closer to each other, so the classifier cannot classify. So that to me, I'm not still sure I understand if this. Why wouldn't this be a correct interpretation?
What you say is perfectly possible, but this is not what the data show. When you try to explain the choices of the animal based on these different mechanisms, the mechanism you propose explain the choice is significantly worse than instead having this amplification because of consistency. So what you say is possible, it just does not... can you hear me? Sorry, yes, it's possible, but it's against the data. So our data suggests that when you have this consistency of information, the readout works better. And then we also had in the paper that I cited in Nature Neuroscience some biophysical explanation for this related to the fact that you have nonlinearities in downstream neurons, and those correlations essentially help the transmission of information through those nonlinearities. But for me, the empirical evidence is that what you say is perfectly plausible. In fact, it's what most people assumed, but does not explain the choices of the animal. I think to explain the choices of the animal, you have to add also this term. So, and so,
Okay, thank you so much.
We were
Another
also
short
surprised,
question.
but okay, yes.
Yes, so thank you very much for the talk. I have one question regarding the negative triplet correlations that you showed in addition to positive triplet correlations. Can you say what is the merit of these negative ones in the concept of information processing thing? And in which kind of behavioral task it is more probable to observe such a correlation?
Okay, thank you, Vin. It's a very interesting questions, also like the one before. So I thank you both, and I'm happy that despite the distance of the problem, we were able to understand the talk, as I understand from the intelligent questions. And so in terms of for which task you are able to see these, we don't know yet because it's difficult to do the experiments, so we don't have a huge amount of different tasks that we did. We have also only the tasks that we tried out, so I cannot give an answer to that question at least yet. But so what is the merit of negative interaction, redundant interactions versus positive interaction, information enhancing interactions? Is that I think the information enhancing interaction just give more information, so they're useful because of that. But the redundant interaction introduce robustness in the system, because if you have redundant information and if a neuron dies, you may have still the information surviving. But also because, as I showed in this talk, they may be also useful for transmitting information downstream. And what this work about labeling the projection and seeing the structures into add to this story is that, okay, you have a bit of both. You have a bit of information enhancing and a bit of information limiting. And then in proportion, we have more one than the other. But given this proportion, if you arrange those in a clever network structure, you can gain more information. So essentially, it's a way to optimize the trade-off of the advantages and disadvantages toward having something that has a bit of both, but with more benefits, even more benefits, because you have more information and still the same redundancy to transmit it. And that's why I think maybe an indication of this is that the design of the neural code is not to encode information locally, but it's to transmit it. So, okay.
Thank you. There is a question from a guest here. He's asking, as we know, the correlation is rate dependent.
Yes.
What about the areas that the rate is very low and during some tasks like working memory in sensory areas? Can in such cases the correlation still be useful?
Thank you again. It's a very good question. I cannot tell much about correlations in areas where firing rates are lower because we did not investigate systematically so many areas. But in our paper, we compared sensory areas versus association areas. And the reason why we did that was that for sensory areas, we thought that the main target is to encode information as much as possible. Whereas for association areas, it's not only maybe you need to encode less information because you can filter out the information that is relevant for doing the task, but you have a higher need of this information being correctly read out to produce the choice because you are closer to the choice. So comparing the transformation in neural codes across areas. With this logic, we found that there was relatively little difference in the properties of individual neurons across areas, but correlation changed, and they were essentially weaker and not limiting information, but also not aiding readout in sensory areas. And they were stronger, limiting information and aiding the behavioral readout in association areas where you have to encode less information. So where the damage of encoding less information is less important, but where the positive of making sure that this information is transmitted generates behavior are higher. So our theory, which we have not proved experimentally, but I think this is a working assumption of our work, is that this trade-off is also dependent, the optimal trade-off on the level where you are in the cortical hierarchy between sensation and decision, and the closer you are to sensation, the higher is essentially the weight in this trade-off toward encoding information, and if you are closer to the decision, the weight is more to transmitting information. So you have to have both, but the trade-off, the optimal level of correlation that results from this trade-off changes across the hierarchy. So this is our assumption, but I think it's reasonable, but I cannot say we proved it because we only consider a small number of areas.
Thank you. Thank you very much for your talk. And if you have more time, there is still a link for a panel that you can attend and people can ask questions there. The link for the panel is written in the chat box. If you have time to, for example, 50 minutes to attend the panel.
Okay.
So the time for the talk is finished, and thank you again for the talk and your time.
Okay, so I will log into the panel. Okay, thank you very much. Okay, thank you all. Thanks to the audience.